Okay, we're going to talk option D, medicinal chemistry. Just give you an overview, we're going to be looking at pharmaceutical products and drug action. Some specific drugs, aspirin and penicillin, as well as some opiates, pH regulation of the stomach, antiviral medications, environmental impact, a drug called Taxol. We're going to delve into nuclear medicine and drug detection and analysis. Okay, here are some objectives. By the end of this screencast, these are what you should be able to do. Okay, let's get a couple of definitions out of the way. First is a drug. A drug is a chemical substance that affects how the body works, and this could be good or bad. Okay, so a drug is going to be anything that brings about a change in the biological function through its chemical action. A medicine, on the other hand, is typically defined as something that treats, prevents, or alleviates symptoms of a disease. Okay, so they have a therapeutic effect or action, and that's the beneficial or the good part about drugs. Okay. We tend to use them interchangeably. Medicines, as we get them, are usually a combination of both active and non-active substances. Okay, and what these non-active, obviously the active is what gives us the benefit, the therapeutic effect. These non-active go to keep the consistency of the drug. Can be the consistency, sorry. Taste. Or the administration of it. How is it going to enter the body? Okay, so administration as well. Trouble with my pen today. All right. The active substance in the medication is going to interact with a target molecule in the body, and that is typically either an enzyme or a receptor or the DNA. Okay. And actually this active substance can do, it can either stop an interaction from functioning. Okay, so stop the enzyme from doing whatever it's doing. From working or functioning. Or it can stimulate it. And that could be the enzyme or whatever the receptor is. Okay. So how do drugs get developed? Well, pharmaceutical companies are in it to make money. And some of the ways that they can make money is if they develop a safer, more effective medicine than a current medication or treatment that's on the market, or if there's a new disease that needs a treatment. And an example of this is when the HIV/AIDS, okay, in the 1980s. Okay, there was a big market to develop something to treat that new disease. You've got to keep in mind that it can take a long time to bring a new pharmaceutical, a new drug treatment to the market, as long as 12 years, and it can cost hundreds of millions of dollars. So what is this first stage of the drug development? Well, the first stage is to identify a lead compound. Okay, a lead compound is going to be a compound that has that desired biological activity. You may find that in an already existing medication. You may have to isolate a compound from natural sources, or you synthesize it from some kind of raw material. So in a process that's known as lead optimization, you're going to synthesize and test a bunch of lead compounds and determine which ones hold the most promise for further development. So these would be either, so you're going to optimize either the most promising or the most active, or sometimes the least toxic, okay, and sometimes a combination of those. Okay, the second stage is toxicity testing, and this is testing for toxicity in animals. Okay, it is in this phase that drugs can have a high failure rate. Right? So you think you've got a medication that works, you get into this second stage, the toxicity testing, and you find that you kill too many animals. Then you cannot bring that drug to the market. Right? And this can be after you've spent quite a number of years in development of a particular drug. After that second stage of animal testing, you go into the third stage, which are known as clinical trials. And this first trial, phase 1, is determining the proper dose for therapeutic effect. And you're also going to identify what the side effects are. If the drug passes this first stage, then you're going to move into the second phase, phase 2. And that's really to determine the effectiveness of the drug. Okay, and this will be on a small number, well, phase 1, let's go back up to phase 1, sorry. Phase 1 is on a small number of healthy people. Phase 2, we're looking at a small number of people who have the disease. And in phase 2, you're going to look at whether the drug is effective in that population, which would be your target population, and again, determine any side effects, positive or negative. Then if it passes that phase 2, you're going to head into phase 3. And this is a larger patient population in which you're going to compare the new treatment with any existing ones. You may also use what's known as a placebo. Okay. And this is typically a double-blind study, which means neither the patients nor the scientists know who had the drug and who didn't, okay, who had the placebo or the existing treatment and who is having the new one. Okay, so in case you don't know what a placebo is, a placebo is something that looks exactly like the real medicine, but does not contain any active drug. you know. Okay, it's made from an inert substance such as starch. Okay, and there is an effect known as the placebo effect. Please spell that right. And that is where people taking the placebo believe that they are feeling better because of taking medicine. Even though the medicine doesn't contain any active drug, there's a psychological response that sometimes people think they feel better, even though there really is no active drug in the medication that they were taking. So if we look at the approximate time frame for developing a new drug, okay, you have about 10,000 possible projects initiated, and you do laboratory and animal surveys on those. That's early in the phase. This is the approximate time in years, increasing as we go down. Okay, and basically the number of projects that move forward gets less and less the more you go through testing. So you start off with quite a bit in laboratory and animal studies, and then when you move on to first phase, some drugs have not made the cut. Going into the second phase, again, some trials or some drugs, medicines don't make the cut. Going into third phase, and then finally the review by the Drug Administration is a small number from what you started with. So let's go into what are some of the drug doses. The therapeutic effect is the desirable and beneficial effect from the medication. Okay, so it either is going to alleviate symptoms or it's going to actually treat a specific disease. Then we have what's known as a side effect. This is an unintended secondary effect of the drug on the body. The ones I think we're used to talking about are the undesirable side effects or the toxic effects. You may also get a beneficial side effect, but it wasn't intended, all right, or that would be a bonus, all right. Some unintended, undesirable side effects are typically nausea and vomiting, headaches, those kinds of things. And the other thing that you should be aware of is Tylenol. Actually has a small range of where the medicine will be therapeutic. Overdoses of Tylenol can be very, very serious. And these can result in permanent liver damage. So if you need to be very careful following the directions for Tylenol, and particularly people should be aware that if you are doing activities that also affect the liver, the next morning you may not want to take Tylenol. Maybe you should think about taking some aspirin. Okay, so determining the dosage. There are a couple of things that we need to define. And one is the LD50. That is the lethal dose of the medication. And this is the dose of the drug that's required to kill 50% of test animals. It's typically measured in mass per kilogram, and that kilogram is the body weight of the animal. Then there is the toxic dose, okay, TD50. And again, this is the dose required to produce a toxic effect in 50% of the test population. Okay, so we didn't quite kill you, but we definitely made you sick. And then there's the ED50. That's the effective dose. This is the dose required to produce a therapeutic effect in 50% of the test population. So interesting, if we look at an example and we take nerve gas, sarin, it has an LD50 of 25 milligrams per kilogram. All right. So if, keeping in mind a conversion factor for every 1 kilogram is 2.2 pounds, what is the lethal dose for 50% of a human population for a 150-pound person? Well, you would take your 25 milligrams per kilogram, and then you have your 1 kilogram for every 2.2 pounds, and you're going to multiply that by your 150-pound person. And this is where you get the 1.7 grams is the lethal dose for 50% of a population of 150-pound people. All right, we're going to look at a therapeutic index, and for the most part, we're going to define it really as this. All right, this is what IB uses, although you should be aware that there are other ways to define a therapeutic index. So the therapeutic index, TI, is the lethal dose for 50% LD50 over the effective dose, ED50. Okay, so say your lethal dose is 100 times larger than your ED, then your therapeutic index would be 100. That's a pretty good number because you have 100 times your effective dose before you get to your lethal dose for 50% of the population. So the higher that therapeutic index, the larger the difference is between the lethal dose and the effective dose. That's pretty much desired, okay? If this is a small number, there's a very small margin of error. Okay? So a low therapeutic index indicates that the therapeutic dose is very close to the toxic dose or to the lethal dose. And you really have to be monitored very carefully. Sometimes you would get this for certain like cancer treating drugs. Okay, so chemotherapy, typically you really have a small therapeutic index, so you want to be very careful how you use that. Okay, so looking at a picture that might better represent this, all right, here we have our therapeutic dose curve, and here we have our lethal dose curve. Okay, a 50 milligram dose is going to cure 95% of the people. That's good, right? Unfortunately, it's going to kill like 5%. Right? So you really have to balance these two in describing and determining the effective dose and the dose parameters. The other thing to keep in mind is over time, you're going to have a window, therapeutic window. Okay, and this is why like every 4 hours you take Tylenol again, because you take it, okay, and over time it decreases. So you take more, it gets you into the therapeutic window, and then it decreases, taking more. You want to be careful that this doesn't pop you up into that toxic. Right? So therapeutic window is the range of the dose between the minimum required to produce a therapeutic effect and the level which produces a toxic effect. These are ranges of doses. Aspirin, and this is a pretty good example to look at. Aspirin at 75 milligrams is used on a daily basis to stop clotting and for heart attack control. Okay, if you're going to use aspirin for pain relief, you're going to need to get 300 to 900 milligrams. All right, so quite a difference in the dose depending on what it is you're using that drug, that medication to do. So the therapeutic window is a well-defined range of the concentration in the blood that gives the desired effect. Okay. One thing to remember is tolerance. The more the body, or the more often sometimes people take a medication, the body becomes less responsive to the drug. So it takes more and more to produce that therapeutic effect. The problem with this is because you have to take more, you're getting closer and closer to that toxic dose. All right. Why does this occur? Well, your body you can increase the metabolism of that particular drug within your system because your repeated use, like as your body kind of gets used to it, knows how to deal with it, metabolizes it fairly quickly. Okay, you're also going to run into some desensitization. You're going to desensitize target receptors. Okay. So the body again adapts, and you need more and more of the drug to make it therapeutic. This can lead to addiction and dependence, and there's two types of addictions. Okay, there's a psychological dependence, and that's the need to have that drug to feel good. And then there's a physical dependence, and the body can't function without the drug. Okay. This dependence is closely related to tolerance. Okay, and it's the need to take more and more of the drug to produce the same effect. That's why if addicts sometimes, if they are recovering and they slip, they sometimes tend to go back to the last level of drug that they took. But what has happened is because they've been off the drug, they don't have the same tolerance for that drug. And a lot of times that's where you see overdoses. All right, so let's talk about administering the drugs. Okay, the majority of drugs are administered orally, meaning we swallow them. Some drugs are going to be administered rectally, and that is through the use of suppositories. And this usually occurs when the patient can't take the drug orally. Okay, maybe because there's vomiting or nausea that they can't swallow it. You also have pulmonary, and those are administered directly to the lungs in the form of gases, sometimes powders, and they go right into the lungs. The absorption in these types is very quick acting because you've got a large surface area in your lungs. Okay, you have topical. This means it is on the skin. Things like creams, ointments, lotions. These are typically for local effects. Things like acne, dermatitis, skin infections. You have transdermal patches like nicotine, and that's just absorbed through the skin. Then you have several different types of injections. Intravenous goes right into the vein. Intramuscular, as the name sounds, it goes into the muscle. Thigh, sometimes the buttock, sometimes the arm, a lot of the time. Okay, and then you have subcutaneous, and that goes into the skin or just directly under the skin. Interesting, this is insulin is done subcutaneously. To give you an idea of what this means, intramuscular into the muscles, subcutaneous right under the skin, and then intravenous into the blood. This is the quickest acting right into the bloodstream. Okay, bioavailability. This is defined as the fraction of the administered dosage that reaches the target part of the human body. If we're talking about administering that drug through an IV, typically it is 100%, right? So the bioavailability of a drug is really going to be dependent on how that drug is administered. Okay. In tablets, some of the factors you're going to look for here is it soluble in water or is it soluble in fats? Is it susceptible to being broken down by enzymes? If it's in a tablet, some of them are going to be broken down and just move through your digestive system as opposed to being absorbed through the intestinal wall. Okay, how soluble it is in water is also going to determine how easily it is absorbed through the intestine. So the more soluble that drug is in water, typically the more bioavailability it has. So we're going to be looking for the presence of polar groups to make it soluble in water, and the size of the organic molecule. Typically very large molecules are not very soluble and have a difficult time being absorbed and delivering any therapeutic effect. All right, looking at drug-receptor interactions. There are things called binding sites, usually proteins found in cell membranes. And there are unique sites, and drugs are produced specifically to fit into the unique sites of the receptor proteins. These medicines should or are synthesized to mimic normal molecules that bind to that receptor. Okay, and they can either make this receptor active or it can block other molecules from bonding to that receptor protein. So what do chemists do in making a drug? Well, one, they make the drug. They design the synthetic process to make the job. They make sure that it's pure through analytical testing and through purification methods. And then they also scale up the effective drug for commercial use. It's one thing to make that drug on a lab bench. It's quite another to make it in a chemical plant and make enough of the drug to be profitable. And throughout the process, they're going to modify that drug for solubility and toxicity, okay, looking at those lead compounds.